Great, thank you, Agatha. And so we'll move straight on to our next talk by Bilal Djeghout, also from the Quadram Institute Bioscience. So Bilal, whenever you're ready, please share your slide. And
okay. Yep. Okay. Okay. Hi. Hello, everyone. My name is Bilal. Thank you for having me here. I'm a postdoc at the Quadram Institute Bioscience in the UK. And today I'll be showing you how I employed a direct sequencing strategy to characterize Campylobacter genomes from metagenomes using human stool. So to begin with, okay, 1 second. To begin with, Campylobacter's genome is around 1.6 million base pair long, with an average gene length of 900 base pair, and more than 94% of the genome codes for proteins, which makes it one of the most densely sequenced bacterial genome. So in diagnostic labs, culture-based testing has been substituted by a rapid PCR-based analysis, which has made Campylobacter genomes unavailable for molecular investigation and epidemiological tracking. Furthermore, the need to preserve information about Campylobacter epidemiology can be achieved by direct sequencing. However, methodologies to this point had many restrictions, including DNA yield volume and precision of downstream analysis. The goal of this work is to improve the approach for identifying Campylobacter species in stool metagenomes. Therefore, the testing was performed on Campylobacter infected individuals. And for this reason, we have partnered up with the regional gastroenteric diagnostic lab to investigate how direct sequencing can be applied in characterizing Campylobacter genomes. For this aim, an experimental method was adapted using criteria for depleting cell-free DNA, enriching microbial DNA, and enhancing high molecular weight extraction to improve downstream whole genome-based sequence analysis. Metagenomic DNA was extracted using 2 different kits from Promega Maxwell and Shoreline biome that included both chemical and mechanical lysine. So Campylobacter cells were quantified in the stool samples using qPCR, and that was to estimate the initial Campylobacter input that is loaded into sequencing, and also to estimate the limit of detection. Then libraries that passed the QC were subjected to sequencing on both Nanopore and Illumina platforms. Generated sequencing data was subjected to in silico analysis, including genome completeness, MLST, and AMR. In parallel, I used this as a baseline to compare. So I grew up to 30 isolates by direct plating of the stool, aiming to do a comparison between metagenome Campylobacter genomes, assembled Campylobacter genomes, and culture-derived genomes. From our metagenomic run, we targeted Campylobacter with the limitations that we knew, and to also know our limit of detection. And to do that, we tested 9 stool samples, where 8 were Campylobacter PCR positive at the diagnostic lab, and 1 was negative, PCR negative. The limit of detection for our purpose was to characterize the species of Campylobacter, sequence type, and AMR profile of known determinant in the stool metagenomes. So what we found was, when we have by qPCR 10 to the 5 cells or higher, we were able to assemble the MAGs and characterize Campylobacter genomes. qPCR was used for quantification of Campylobacter, and to assess the quality, we used fragment size in the library prep that had a direct impact on the quality of the reads. So we did not find an association between the total number of reads and the ability to map Campylobacter genes. What we found here is that qPCR can give us an idea of the quantity of Campylobacter in the metagenome and what to approximately expect from the sequencing. And as you can see in this table, the amount of Campylobacter reads that was generated from the sequencing ranged from 1,000 to more than 400,000 reads. Samples highlighted in bold, I was able to assemble sufficient Campylobacter genomes and analyze them, and I will be talking about that in the following slide. To highlight the effectiveness of our method, in 3 stool metagenome samples, Campylobacter genome was assembled to more than 90% of a complete genome. Those samples contained sufficient read assemblies for characterization. Therefore, a comparison of MAG-derived and culture-produced genotypes revealed that of the 3 MAGs, 2 Campylobacter jejuni and 1 C. coli were detected. C. jejuni genomes derived from direct sequencing were classified as sequence type 61 and 21 respectively, with resistance gene BLA-OXA61 in both. And as you can see in this table, we saw matching species and sequence type between MAGs and culture-derived genomes. We did see a slight difference in the resistance profile where the completeness of the genome was lower. We also noticed a difference between genomes and MAGs about the number of known virulence genes. So this is one of the aim of this study, is to be able to say that we can characterize Campylobacter genomes from a stool specimen using direct sequencing approach. This graphic represents reads that were classified as Campylobacter in one of our metagenome samples when the patient was infected with 2 different species of Campylobacter in the same time. We found that in case of co-occurrence, higher quantity, quality, and depth of coverage is needed to characterize both species in the stool metagenomes. You can see, for instance, that we detected the 2 species, but we were not able to get their marks. In conclusion, this study demonstrates the limit of detection of Campylobacter within a metagenome and the ability of tailored direct sequencing methods to yield sufficient information for near complete Campylobacter genome assembly to characterize key attributes to this foodborne pathogen. So for now, we are working on processing and post-processing techniques refinement to improve the quality and amount of direct sequencing information produced by both short and long read sequencing to target Campylobacter. Okay. I would like to thank my group leader, Dr. Nicole Giannico, and everyone involved in this work. And thank you for listening.
Thanks, Bilal. So we've had quite a few questions come in. We'll get through a few. So first one actually from Ankatrin says, you probably said it, sorry if I missed, but what sequencing strategy and what depth did you have? Perhaps you could just remind us.
So here, sorry, for the sequencing strategy, I used short reads and long reads. And before that, we had like quality control of the library prep, and we had like certain criteria before putting that metagenome into sequencing. For instance, the quantity of Campylobacter in the metagenome, if it's enough, and also the fragment size and yeah, and that's it.
Okay, and next question from Nabil, another technical one. How to actually constructed, what software was used, and what was the average length of the contigs?
So for the average, I mean, I don't remember exactly like for each sample what was the average length. So yeah, as you can see in this table, like there are plenty of numbers to remember. But yeah, in general, for short read, there were no issue with the fragment size. yeah, the only issue we faced was with long read sequencing. That's where
Okay.
we were not able to assemble the food genome of Campylobacter.
Okay, so there's a follow-up question from Alda about different sequencing approaches. There's also been a question on how do you deal with plasmids with your megs? Quite a few Campy have plasmids. How do you deal with those?
So for this study, for now, we were only interested to know the sequence type using MLST and the species definition and the genome completeness. So further analysis will be done later to explore or instead to do more in silico analysis. So for now, I focus it only on the MLST, AMR profiles, and the species.
And finally, a quick question from Brendan. What do you define as a virulence gene in your table?
Sorry, can you repeat that question, please?
What do you define as a virulence gene?
So virulence indicator genes are, for instance, the genes implicated for the adhesion and also for the motility and so on. Yeah, we have like more than 100 virulence genes.
Okay, thank you.